The subjective evaluation was set up to test two hypotheses regarding voice identification by humans. The first hypothesis assumes humans do more voice identification in a narrow set of speakers than real voice verification or identification. They operate with a discriminating group classification. They separate the speakers relatively to the others by group. The second hypothesis requires many binary tests to perceptually assess the human ability to perform voice identification in order to shadow the consequences of the first hypothesis. However, a fresh voice in a binary test is not equivalent to a voice exposed in another binary test and implies memorization bias. Therefore, we propose a configuration where the listeners must perform a voice clustering task, and we hypothesize that this configuration is more economical in terms of number of trials. In addition, this task may be more ecological than a binary task. How do humans cluster speakers? A web interface was developed where recordings are presented to the listeners as numbered points. Listeners are asked to classify the recordings into groups corresponding to different speakers. Some of the recordings were anonymized, which affects the voice quality. Speech recordings were selected from the VCTK corpus, which is composed of English sentences read by native speakers. 15 female and 15 male speakers were selected and divided into reference and distractor groups. 24 utterances were selected and reduced to maximum duration of 3 seconds. The reduced recordings are called chunks. A session is composed of 3 trials. Each trial is composed of 16 different chunks distributed between 3 reference speakers at the rate of 2 to 6 chunks per speaker and 1 for the distractor. The control trials contain original chunks only. The evaluation trials contains 8 anonymized chunks, including the distractor's chunk. All anonymized chunks have been processed by the same system. The 39 listeners include 13 women and 26 men. Most of listeners were native French speakers, and 9 were native English speakers. Here are our results based on our listeners.
A 2-tier analysis was conducted. Tier 1 examined the effects of anonymized recordings mixed with the original recordings over the course of the trials. Tier 2 looked at the effects between the anonymized recordings between systems during the evaluation trials. Several response variables were used in our ANOVA: the listening duration and the number of times spent listening to speakers, the time spent moving the points in the interface, and to measure accuracy, the F1 measure score was calculated by identifying the centroid of each cluster. The mean and standard deviation for each variable was used. For Tier 1, while we observed no differences between trial types, we observed significant differences between speaker genders. In general, when presented male speaker stimuli, participants had higher average F1 scores with lower deviation, lower average number of listens, and spent about a minute less on average when compared to the female speaker stimuli. For Tier 2, we observed a significant main effect on average F1 scores between baseline systems, with participants performing better with baseline 1. With the same system, participants performed with lower variability on move duration. However, we report no significant correlations between these metrics. Finally, we observed a significant main effect on average F1 scores between speaker genders. However, unlike Tier 1, we found participants performed with higher accuracy when presented female speaker stimuli.
We also use what we call the clustering homogeneity measure, which corresponds to the ratio between the number of chunks in the corresponding clusters and the total number of chunks, which is expressed as a percentage. The tables present the distributions of homogeneity measures for female and male speakers. As we can see, the clustering homogeneity measure is higher for the control trials, which is expected because they don't contain any anonymized chunks. We can observe no significant difference between male and female speakers in terms of clustering homogeneity measure. For both gender, the Kruskal-Wallis test shows a significant effect of the type of trial on the homogeneity measure. P-value is lesser than 0.001. The Mann-Whitney test between both baselines show no significant differences in terms of homogeneity measure. P-value is superior to 0.01. Thank you.